Greetings everyone, I'm Froneman and today I'm going to be giving you a brief overview of my research project. The title of my first presentation is Assessing the Role of Ascospores in the Epidemiology of Apple Scab. Apple scab is caused by the fungus Ventura inequalis, which overwinters as ascospores in fallen leaves on the orchard floor. In South Africa, ascospores have always been known as the main source of primary inoculum, causing the first infections in spring. It is also known that in orchards with pygmy fruits, infected pygmy fruit, canidia from the fruit lesions can also cause primary infections. So the main question being asked was, is the role of ascospores changing in South Africa due to climate change? In the past 5 years, we experienced warm and dry autumn and winter conditions, which were not conducive for ascospore formation. The analysis of 10 apple scab orchards over the past 4 seasons showed almost no ascospores in spring on infected leaves overwintered on the orchard floors. Also, very low numbers of ascospores were caught on spore samplers during the seasons. The low ascospore presence could be the reason for the low apple scab incidence in most orchards during the last 5 years. However, there were a few orchards that had low to medium scab severity. So a second question arose, could canidia have initiated these epidemics? Therefore, the aim of the project was to determine the relative importance of ascospores versus canidia in the epidemiology of apple scab. Two objectives were needed to reach the aim. The first objective being determining the presence of viable canidia on leaves overwintering on trees. And the second was to evaluate the role of ascospores versus canidia in spring during the past 2 seasons using a population genetics approach. So for the first objective, leaves still attached to the trees after the winter were collected and used to observe germinating canidia to determine their viability. It was determined that canidia can survive the winter on leaves that remained on the trees, and that post-harvest EBI sprays reduced the number of viable canidia by only 20%. For the second objective, population genetics was used to evaluate the role of spore types in spring infections. Orchard trees and potted trees were used. On orchard trees, the first leaf lesions over 2 seasons were sampled at multiple locations within orchards, and the lesions will still be genotyped. Potted trees were placed within orchard work rows and exchanged after each rain event. The first lesions in the season were collected and genotyped. So the genetic analysis of the potted tree lesions showed that clones occurred only in an orchard with high fall infections, indicating clonal infections. And the signal for sexual reproduction was seen on all potted tree populations, which is an indication of ascospore infections. So future work will involve a similar analysis of the orchard tree populations and the determination of whether orchard populations differ from potted tree populations, which would be an indication of canidial infection. Conclusions that can be taken away from this project thus far are that in South Africa, canidia can cause some of the first scab infections in spring in orchards with high autumn infections. At the end of the project, more data will be available to make a final conclusion. Another conclusion that can be made is that canidia can survive the winter on leaves still attached to trees. This shows the importance of chemical leaf drop approaches in late fall and spring. Other studies have been done and shown that canidia can also overwinter in dormant buds in other regions of the world with moderate winter temperatures, especially with high autumn infections. So this could be a future study to determine this in South Africa. The members of the project team include myself, Alzane, as well as Celeste Linde, Professor Adele McLeod, Dr. Lindani Moyo, Trevor Koopman, and Becker Wessels.